yesterday I planted some Lepidium sativum seeds, cress seeds, in these little microcentrifuge tubes, and as you can see right there, and so they had to sit in the fridge for 24 hours, and now they're ready to go. And one last step I got to do is I'm going to actually cut the top off of this. If you imagine the centrifuge tube, and then it has a top on it. Well, I've filled it with agar, and then I've planted a seed in there. And then now that it's been in the fridge for 24 hours, I'm actually going to cut this section off. That way it can actually grow and sprout. And the idea is they're like little planters that I can grow the plants in. And then what I'm going to do is I'm going to put it in the incubator, which is this thing in the back here. Now that incubator, it has a 24-hour light cycle, grow light cycle, and it has temperature control of up to 30, or well, I can set it to, well, theoretically whatever, but obviously there's limits. But I have it set at 30 degrees Celsius, and it also has aeration access, which is I don't know if you can hear it or not, but there's air lines that go in there, and I can put up to 6 different samples in the incubator. And it, like, right now there's algae in there, and there's actually an air line that goes through that into the beaker that contains the algae, and it aerates it, and it provides carbon dioxide, and it provides motion to the algae, because the algae needs to stay in motion. And the incubator, it's interesting. It's actually a fish tank turned on its side, and then there's a PVC or not PVC, plexiglass put across the front, and then I put 2 holes where I put these gloves in here. And the idea is so that I don't have to keep opening the incubator all the time just to do something in there. Obviously, if I have to put stuff in there, I can do that, but if it's just manipulating stuff, because I also have a pipette in there, wash bottle, Kim wipes, parafilm, some extra water, petri dishes, sanitizing alcohol, isopropyl alcohol, and a whole toolbox full of tweezers and all sorts of stuff to be able to manipulate whatever samples I have in there. It actually this is an air filter which goes through here into here to be able to it's a HEPA filter, and it filters out all the dust and impurities, because obviously this is a garage, so you're not, no matter what, you're going to have some kind of dust collection and so on, and of course you try to clean everything as much as you can, but in here it filters out that way. Now this glove hole actually comes off. There's 4 wing nuts that I can take off, and that's how I'm going to put the seeds in there. Every once in a while, the reason the alcohol is in there is so I can sanitize all the surfaces every once in a while. But one thing that it also has, so I don't have to go through this process all the time, is when I do my algae counts, there's actually like a little place right here, little thing that opens up, and I can send the sample through there without opening like all of this. I only need to use this if it's a large sample that I need to bring in or out. So I place it in there and close it up. So now I got to place it underneath the grow light. And to do that, I have to take off my lab coat, because I can't get my gloves in there when I can't get my hands in the glove in the gloves with my lab coat on. Now, of course, I got to make sure to not knock everything over in there while I do this. One thing I got to do is since I'm in here, I have to do my algae count, which is essentially I count the algae cells, and I record it. And since I'm messing around in here, and it's around that time anyways, I might as well do it. Now, working with these gloves can be quite tricky because you're talking about these little tiny pipette tips and tiny centrifuge tubes. So what I do is I take the pipetter and I go into the sample of the algae sample, and I remove 100 microliters, which is a tenth of a milliliter, and I put it into a centrifuge tube. Now I have a little PVC pipe, which you'll see in a minute, which I use specifically to transfer samples out of here without having to open up the big door. And then now now I open up the tube here, and I have the sample, and in there as well is the centrifuge tube. As you can see, it's a PVC pipe, pretty simple thing. I melted the end of it. That way I don't have to open it up. And so I'll end up doing that count, which I did a video on that a couple days ago, I believe. So you can check that out. Other than that, thanks for tuning in.